One of the biggest questions that's now really been quite well answered by evolutionary biology is, why do we age? Do we have to age? Do we inevitably grow and die? Here you can see the process of aging indicated for a human male. It's fairly dramatic, and we're all quite familiar with it. However, it is underpinned by a long evolutionary history. Biologists define aging as the decrease with age in the intrinsic ability to survive and reproduce. If a person gets killed in an accident at age 78, we don't count that as aging unless some particular condition brought them quite clearly into the only possible way of having that accident. Aging is intrinsic to the organism and therefore has to be measured carefully in environments where other factors are ruled out. We see aging as a byproduct of selection for reproductive performance. Aging in and of itself didn't evolve as an object of selection. It evolved as a byproduct of selection. It explains our susceptibility to degenerative disease like cardiovascular disease, like type 2 diabetes, like cancer, like Alzheimer's, and it explains why we must die. A general conclusion that you will see in a few minutes is that all organisms that have asymmetric reproduction must age. And by asymmetric reproduction, we mean at some stage in their life cycle, there is a division of a cell where one cell gets the new parts and the other cell gets the old parts. That's the source of the asymmetry. Now, there are several parts of this argument. One is the selection part, and we'll now take a look at how selection varies with age. Selection on survival decreases in intensity with age. Peter Medawars was the first to really articulate that. He was a brilliant Brazilian, British immunobiologist who got the Nobel Prize for his work on self-recognition in the immune system. And in 1952, he said, the force of natural selection weakens with increasing age, even in a theoretically immortal population, provided only that it is exposed to real hazards of mortality. If a genetic disaster happens late enough in individual life, its consequences may be completely unimportant. Well, that's interesting, but maybe not completely clear yet, and so let's unpack it a bit. Visualize the distribution of ages of your ancestors at the time they gave birth to your lineage. So your mother's, your grandmother's, your great-grandmother's age, and so forth, going back. It's dominated by the young. It's not dominated by the old. Medawars saw that even if organisms don't age, there are going to be fewer old than young because there's always some mortality. And that mortality, at least initially, occurs in organisms that aren't aging. So let's suppose we had a population of theoretically immortal humans who were simply experiencing the average accident rate that humans have. They get hit by tree limbs, they're in car accidents, they're in things that are not associated with their age. Well, there are fewer of them when they are old than when they are young, simply because accidents have happened. And that means that there will be fewer opportunities for genes to pass through older organisms. And that means that younger organisms are more important to selection. Because evolution's a numbers game. Selection is stronger on events that happen frequently, that is reproduction by the young, than on those that happen less frequently, survival in the old. So if the whole point of evolution is reproductive success, it has these consequences for people of different age. Now, that's the selection component. That's the way the environment is affecting things. What about the intrinsic component, the design of the organisms? George Williams added an important genetic effect called antagonistic pleiotropy. Antagonistic pleiotropy means that a gene improves organisms early in life and it makes them worse late in life, in some respect. Mutation accumulation is another possibility. That would mean that early in life the gene doesn't have any effect, it's just neutral, but late in life its effect is negative. Both of these kinds of genes can accumulate in populations, but most of the evidence suggests that we are loaded with genes for antagonistic pleiotropy, and it's uncertain whether there's been very much of this kind of mutation accumulation. Okay, now, what affects selection on lifespan? What would happen in the environment that might cause us to evolve to live longer? Well, changes in selection that increase the relative contribution of adults and decrease the contribution of juveniles or the young. That would mean lower adult mortality rates, higher juvenile mortality rates. So if one moved into an environment where adults were essentially free of illness, but infants and juveniles suffered from a lot of infectious disease, that would select for longer lifespan. In contrast, if you wanted to ask, well, what would make us be selected for shorter lifespan? If adult mortality rates increase, then organisms should evolve more rapid aging. And there's an easy way to think about that. Why invest in maintaining a body that's going to be dead anyway for another reason? It's better to concentrate on reproducing. So that, in English sentences, is an approximation of a lot of equations in life history evolution. So what are the causes of aging? Well, as said, it's a byproduct of selection for reproductive performance. It arises through the accumulation of many genes with positive or neutral effects on fitness early in life and negative effects on fitness late in life. Now, because we are bundles of genetically variable trade-offs, and because aging is a byproduct, and because patients differ genetically and developmentally, we expect aging to have many causes that differ among individuals. People grow old in many different ways. If we then fix one problem, another would emerge. That appears to be true. First, we reduced mortality caused by infectious disease with vaccines and antibiotics, and then cardiovascular disease emerged as a major killer. Then, we improved the prevention and treatment of cardiovascular disease, and now patients die frequently of cancer and Alzheimer's. It's not yet clear that this process has an end. Now, Tom Kirkwood developed William's idea with what he called the Disposable Soma Theory. Basically, Tom was trying to see whether he could connect the molecular mechanisms of maintenance with the evolutionary ideas about aging. And on the y-axis, he drew investment in repair. So this would be how much you put into all of the different repair mechanisms in cells. And on the x-axis, he drew fitness. And what he saw is that at zero investment in repair, the organism would fall apart right away. Then as you invest more and more in repair, its lifetime fitness goes up to a certain point. But if you invest more than that in repair, fitness starts to drop off because you're investing so much in repair that you're compromising reproduction. And because of that compromise with reproduction, he saw that the level of repair that would be necessary to keep you alive forever, to be potentially immortal if you don't have an accident, would always be to the right of the amount of repair needed for optimal fitness. Okay, so that's why he called us the disposable soma. We are disposable because we get used up in order to reproduce well enough to get more copies of our genes into the next generation. Now, what kinds of organisms should age? Well, Auguste Weismann, who was a brilliant 19th century biologist, said in 1882 that asymmetrically reproducing organisms would be the ones that would age. And he thought at that point that there were some examples of immortal organisms that divided perfectly symmetrically that did not age. That has since been shown to be wrong. George Williams thought that it was sexual organisms that had a separation of a soma, which was mortal, from a germline, which was immortal. John Maynard Smith thought that sexual organisms that returned to a single cell stage should age, and that allowed him to include plants, and plant senescence is well documented. Linda Partridge and Nick Bart went back basically to Weismann's definition and said any asymmetrically reproducing organism in which the mother can be distinguished from the daughter should age, because the mother, who had already reproduced, would have the old parts and would not have the probability of survival of the daughter, who was younger and who had the new parts. So if we look at this in bacteria, we would say, okay, symmetrically reproducing organisms shouldn't age, because when division is perfectly symmetrical, it's impossible for selection to distinguish a mother from a daughter. There is no mother or daughter. There are just two cells that have been produced. Both are equally intact or equally damaged, and the reproductive payoff from improving the maintenance of both of them is equal. So there wouldn't be any distinction then between the older and the younger, and that would mean that neither of them would be selected in a way that would cause them to age. They'd both be selected to be maintained. Well, let's take a look at a bacterium that reproduces asymmetrically. This is Caulobacter, and basically its life cycle goes like this. The mother is sitting on a stalk attached to the surface. She divides, producing an offspring, which swims off like a larva. It sits down, and then it goes through a developmental period until it reproduces. So this is a period from birth to maturity of development, and then it's into a reproductive period, at the end of which it dies. So in some ways, it actually looks rather like a multicellular eukaryotic organism. Does Caulobacter age? Well, take a look at the age of the stalked cells and their reproductive output. It declines, and the rate of decline is increasing with age. So here is a bacterium, which were not thought to age, which is aging, and it reproduces asymmetrically. What about something that looks like it really reproduces symmetrically? Escherichia coli, the common intestinal bacterium. Well, if you label the parts of the cell, you can distinguish an old pole from a new pole. Remember that the new pole is going to be in blue and the old pole is going to be in red. And because of that, you can have cells that had a new pole in this division, and then had a new pole in the next division, and that then had a new pole in the next division. So they can go new, 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 or they can go old, 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 and you can have everything else in between. And if you follow those cells, this is the effect of that asymmetric division. So here was the original cell, and then this is how long it took the new pole to divide and the old pole to divide. This is new, new, new over here, old, old, old over here. And by the time you go through 1, 2, 3, 4, 5, 6, 7, 8 divisions, you can see that the cells with the new pole are actually producing better than the cells with the old pole. This is their growth rate. This is not their division time, this is their growth rate. And if we normalize their growth rates, then this would be the growth rate of the new poles, and this the growth rate of the old poles as they go through these consecutive divisions. The new poles are growing more rapidly than the old poles, and eventually the old poles will simply drop out and disappear, and we will be left with the offspring of just the new poles. These are senescing, they are aging, and these are being renewed by division. So that indicates that the criterion for who should grow old and die is basically anything with asymmetric reproduction, which appears to include everything living as we know it, with the possible exception of viruses. What are the mechanisms that mediate aging in organisms like us? Well, energy flow makes protons as a byproduct. They leak out of mitochondria when we process our food, and they cause oxidative damage. If we increase growth rate and therefore increase metabolic rate, that causes energy flow to increase, which causes more metabolic damage. The candidate genes mediating growth hormone, insulin, and insulin-like growth factor are all genes that are thought to influence aging. If you target mutations in these, or you analyze spontaneous mutations in these genes in mice, there are variants that increase lifespan up to 70%. And there are similar effects that have been observed in flies and worms. Now, humans are neither mice nor worms. And the mutations in these pathways in humans don't lengthen life, except possibly in one cluster of dwarves that live on an island in Croatia. And they are prepubescent, the size of a 7-year-old, and chronically cold and sleepy. So it's not exactly the kind of germline therapy that you would want to carry out to increase lifespan in humans. It has a lot of side effects. So what would happen if we selected humans to live longer? This would probably be the best way to do it, because that would mean that all of their physiological systems would be improved. Could we do it? Well, the heritability of lifespan is about 0.25. That's enough to work with. So there's genetic variation in humans for lifespan. The response, if there were strong selection, would be rapid. It could act by delaying age at first birth progressively. So, for example, we could all agree that we would not have children until we were 30, and then after lifespan had responded to that, we could then all agree, okay, now we won't have children until we're 35, and so forth. And that would mean that people would be designed to be in great shape up until they started reproducing, and that could just continue to progress. All of their systems would be improved. They'd be healthier and more vigorous at ages that are currently thought to be very old. You would eventually get 70-year-olds that had the physiology of a 17-year-old. The upper limit would probably be reached at several hundred years, and that would be the expected lifespan if all intrinsic sources of mortality were eliminated and everyone eventually died in accidents. Now, one comment is that this is a fantasy. It could never be implemented because many people would cheat and reproduce early. And the other is that this may be a case of being careful what you pray for, because it is not clear that we would be happy at the age of 200 or 300. So to summarize, aging evolves in any organism that reproduces asymmetrically, and that seems to include all organisms. It does so for two reasons. One is that selection intensity declines with age anytime a mother can be distinguished from a daughter. Genes that improve early performance at the cost of late survival cannot be kept out of a population. They are selected for and they will accumulate over time. That's antagonistic pleiotropy. That means aging is not directly selected. It's a byproduct of selection for reproductive performance. This idea that I've described has been repeatedly tested in many organisms, and it has usually been confirmed. It seems to be very solid. That is why we grow old and die.